Okay, folks. If you don't mind, I'm going to try to get started. We'll see how things go today. A couple of other things that I wanted to mention. This is actually a very interesting class in the sense that there's a fair number of you who are neuroscience majors or specialists, and then there's another group of you who are in the applied functional genomics. And so I'm going to talk to you sort of from a standpoint, sort of in the middle ground, where I'm not going to talk about too much neuroanatomy, nor am I going to go into too much detail about specific elements of genetics unless we're learning a technique. But if there are any questions along the way, especially with the models related to epilepsy, not necessarily today, but in the next lecture, I want you to let me know, because we are going to go through classic models in epilepsy, and hopefully after today's lecture, you'll understand the reasons why we're going to be covering those. I want to actually take a step back and talk to you a little bit about why you actually learn neuroscience and why you actually learn genetics. And the rationale for that might be something that you haven't thought about, but every course that you take here at U of T or anywhere related to neuroscience is actually about this. It's about understanding circuits. So if you're taking a neuroanatomy class, you're learning neuroanatomy not to learn about where different structures are, but how those different structures lie in relation to another structure and how they communicate with each other. When we talk about synaptic plasticity, learning, memory, disease states, actually what we're really looking at is how that circuit is supposed to work and what's broken about that particular circuit. And so I want you to change your focus today just a little bit, because if a psychiatrist and if a neurologist is looking at a circuit in the same way that I've just explained to you, then as neurobiology students who one day might have the goal of becoming a psychiatrist, a neurologist, a neurosurgeon, all of them, by the way, look at the brain the same way. It's a circuit. Then I want you to maybe think about how you might alter the way in which you approach your neuroanatomy, the way that you approach different disease states, and whether or not the vast majority of what you learn, whether it's genetics and understanding the genetic basis for different diseases and how those circuits become corrupted, I want you to start thinking about that in a different way. And you can look at whatever you want in terms of genetic techniques, behavioral studies, all of those, every last one of them really fundamentally goes back to what is the circuit that underlies whatever we're studying, okay? And I'll give you some examples as we go along. So this is one of the real reasons why we actually study electrophysiology. It has nothing to do with learning about the resting membrane potential. It has nothing to do with action potentials. It's getting one signal from one part of the brain to another, from one part of the circuit to another. And that's really why we learn all this about electrophysiology. Again, genetic studies, how we identify disease populations, it's to understand sort of the underlying circuitry, especially related to the brain. When you start learning about the embryological development, really what you're learning is how those circuits get set up in the first place and what happens if those circuits misfire or are not properly connected. So we're going to take a little bit of time and understand some of the roadblocks, and then we're going to look at one of the techniques that has recently come out by a psychiatrist called optogenetics. This technique is now 11 years old. This technique is now considered a standard technique across neuroscience, and I want us to understand it, and I want us to use this sort of in a novel way to understand different types of circuits, just like Carl Diceroth and others have used this as well. So I'm going to give you some of the basics. Some of it will be a review for some of you, and for some of you it will be brand new, but I am also going to give you a little bit more depth if you've learned about this before with me in other courses. This is the way a neurosurgeon or a psychiatrist or a neurologist looks at your brain. Each of those structures, those boxes that you see there, are structures that you can identify if you've taken a neuroanatomy course. So many of you will know this classic circuit that is related in green to Parkinson's disease, but it really is literally a circuit. And this circuit that is in Parkinson's disease, we know that there are important components to it, things like the subthalamic nucleus. We know that the globus pallidus, both the internal and external capsule, are important parts of this circuit. But we know that without this circuit, that movement and or abnormal movement comes about as a result. Whether we look at the motor cortex and the premotor area or the supplementary motor area, it's really all a circuit. And this is the way that we try to understand the brain. The big issue in neuroscience is we simply don't know how all these circuits line up. We only know where the boxes are. And we think we know where most of these boxes are. But if you look at the last 25 years of neuroscience, which has been the biggest era of neuroscience over the last 200 years, the last 25 years has actually done more for neuroscience than the 175 years that preceded it. If we take a look at all the things that have been done in neuroscience in the last 25 years, it's really to define these circuits. Whether you're thinking about a neuroimaging method like microscopy, whether you're talking about clarity, what you're really doing is trying to see what the underlying circuits are. So right now, if you're thinking about this, you should be thinking, how could I, as a neurobiologist or a neuroscience student, help to understand these circuits in some more detail? So these boxes, for example, are pretty well established groups of neurons. We know what they do, we think, but the wiring between them and the whole circuit and what happens if you yank out parts of the boxes are things that we're still coming to terms with. So our current understanding of the brain comes from a number of different studies. So some of you will have had these in psychology, for example. It's a classic study that I'm going to go back and revisit with all of you in the next few minutes. And this is a lesion study, very typically done where you damage an area of the brain, or you could have a brain tumor, or you could have a cutting of axons. Whatever it is, a lesion in an area of the brain will often tell us how that fits inside the circuit and what the normal behavior might be. We know now from a number of autopsy studies that there is a structural basis and an anatomical basis for how these different areas communicate with each other. And we know, for example, during different types of neurosurgery, which is why neurosurgeons are incredibly interested in this circuit diagram, that through brain stimulation, turning on one group of neurons, one of those boxes in that previous slide, that you can elicit certain types of behavior, especially during awake procedures. We know that much of the function of the brain comes from brain imaging studies, and we know that especially nowadays, when we are looking at things like MRI or structural imaging, or whether we're looking at receptor binding through positron emission tomography, or we're looking at white matter tracks, all of it is really designed to either tell us about those boxes or about those little wires that connect the boxes together. So going back to classic neuroanatomy, and going back to classic studies, and I don't want you to memorize this. This is something that you've all had in first-year psychology or even in high school. This is Phineas Gage, as many of you will know, and Phineas Gage had a horrific accident that damaged his orbital frontal cortex. And again, what this illustrates is that through damage to an area of the brain, the orbital frontal cortex, we know that you can change someone's personality, the way that they act, decision-making, and other types of things. Then post-injury, he was a very, very different person than he was before he had this injury. We know from reconstructions that were done based on his skull exactly the type of lesion that he probably had. We know the size of the lesion that he probably had through inference. And we know that his personality, based on writings that are still available, that Gage's personality changed completely after this particular accident. And again, this is sort of the orbital frontal cortex. So if you're thinking about your eyebrow ridges, this is the area of the brain that's sitting right above and behind your eyebrow ridges. This is the area that was damaged in Phineas Gage during that accident where he was using live gunpowder, live exploding powder to use a rod to tamp it down. And again, this is the area of the brain that we get a lot of information from lesion studies. So this area of the brain, by the way, has a number of issues associated with it. And I'm going to focus on this just to give you an idea of the types of circuitry that we are looking at. For those of you who are taking, for example, the neuroanatomy course, this might mean a little bit more. And for some of you who haven't, you don't need to worry about understanding the circulation. But we know that this area can undergo damage for a variety of reasons. We know that you can have a cerebral artery stroke in the anterior region. You know that you could have a meningioma. You could have damage or blood vessel expansion in the cribriform plate in that region. For whatever reasons, this area is very vulnerable to damage. Okay, when these types of things happen, you can have all sorts of changes. They don't have to necessarily result in the same changes that we saw with Phineas Gage. It might actually lead to decreased aggression. You might have a feeling of euphoria. You might not be able to feel things, which is the lack of affect. You might become more irresponsible, or you might become more impulsive. And again, one of the reasons why I really like this slide, I don't know if any of you know Antonio Damasio. Have any of you heard of Damasio before? It's really big in neuroscience and specifically in psychology. Antonio Damasio. He's actually a U of T grad before he moved down to the United States, and he worked with Derek Vanderkooy, who's still here at the University of Toronto, and he came up with the Iowa gambling task. And part of the Iowa gambling task is related to him trying to figure out in individuals that have damage to this area of the brain, why do they start taking more risky behavior? And this is sort of the Antonio Damasio that you might have heard about before. We know that this area, because if you go back to that circuit diagram that you don't have to memorize, actually feeds into a number of other areas. And so memory is affected in individuals that have damage to this area of the brain, which you might find surprising, that damaging the orbital frontal cortex actually might affect working memory as well as visual associative memory. So this tells us that a lesion to the brain actually probably impacts on multiple areas. In fact, there are probably connections that we aren't aware of that based on these types of studies that lead to us guessing at this point, because we have no firm evidence, that these areas might be linked up in a number of different ways. Now, another area of the brain, which is the medial prefrontal cortex, and in particular the ventral medial prefrontal cortex, because it's a larger area, often is mistaken for the orbital frontal cortex. Again, lots of reasons why we might have damage here. So you might have traumatic brain injury. You might, simply damage yourself. You might get hit really hard on that area of the brain, and you might damage this particular area that you see here outlined in red. You might have lesions that lead to changes in your intelligence, etc. So all of these things tell us that these circuits actually have profound effects. And by the way, later in this course, if you actually believe any of this is true, that we're actually looking at circuits, so I'm going to try to convince you when we looked at earlier, and if some of you took HMB300 with me before, whether you're talking about depression or you're talking about things like bipolar disorder, anxiety, all of those are circuits that are malfunctioning. But do you think intelligence actually involves a circuit? Because we are going to come back and examine intelligence from a genetic standpoint. And is intelligence actually a circuit, or does it go beyond sort of this sort of notion that psychiatrists, neurosurgeons, as well as neurologists have, that everything sort of outlines or has unique pockets that have things related to wiring between different areas? And we're going to take a look, and we're going to see if this theory is actually going to stand water when we look at something that we can measure, like intelligence, in a genetic system. But I want to go back and revisit this, because this becomes important in our discussion of things related to temporal lobe epilepsy. And again, I'm going to be jumping around all over the place, and I'm going to tell you that the types of things that you should take away from today's lecture are not really the trivial details about things related to Phineas Gage. I'm not even going to ask you to know a lot of the details of the slides that are here. It's more to try to figure out how much neuroscience you know. So this is a case study from one of my colleagues over at Toronto Western Hospital. And he had sent me these slides a number of years ago. And this is an MRI, as you can see. B and C are an MRI. I'm not showing you A. It's not really necessary to see the size of the lesion that he has there. This is a 40-year-old male who was otherwise healthy, had a normal marriage as well as family life. And during the course of 1 year, he started to engage in behavior that he had never engaged in before. Okay. And during the course of this 1 year, he started to actually engage in a type of behavior that society, culture, everything says is against the law. It's something that wasn't part of his normal behavior, and he started to collect child pornography. He knew that this was wrong behavior, by the way. He concealed this behavior from his family actively. So his rationality, his reasoning skills were still intact. However, the police became involved once they were aware of his activities. He was ordered out of his home because he had young children at the time, and he was ordered by the court to be prescribed a form of progesterone. Do any of you know what this is used for, this type of progesterone? Medroxyprogesterone. Have any of you heard of this? No? So have any of you heard of chemical castration? So this was a serious offense. This was an individual who had a lot of changes in behavior over the course of 1 year. He had no overt damage, but what ended up happening is because he started to have other complaints where he had complaints of headaches, he started to have balance problems, an MRI was ordered, and this is sort of what came up on his MRI. So those of you who are taking neuroanatomy, and again, you don't need to worry about the details here, what is being affected? Or what do you think is being affected? You can see this large sort of mass that is growing here. What area of the brain do you think this is? Just to orient you, this is the front, and this is the posterior region of his skull, and this is sort of in a sagittal section of his brain, as a coronal section of his brain as well. What do you think is being affected? And you can see it's a large tumor. It grew incredibly quickly over the course of 1 year. Any ideas? Yes. So this is part of the ventral medial prefrontal cortex. Different areas were also affected. So luckily for him, and again, with him, we also saw behavioral changes in this area of the brain. The orbital frontal cortex and the medial and ventral medial prefrontal cortex are not identical, but they overlap in many regions. And so there were changes in his behavior, there were changes in the types of responses, his personality, etc. So you have to start thinking, like, why did all of this occur? Again, if you're thinking about this, if you go back, and I'm not sure if, I don't think I have all of the details here. What do you think happened with that individual? Yes.
Could it be that the changes in personality due to the tumor of his...
Absolutely. So he had a tumor, and it was affecting his personality by changing the circuitry. It was pushing the circuits to the side in the medial prefrontal cortex. What do you think was his treatment? They didn't just let it keep growing, right? So this becomes one of those things. In the brain, you have a number of different options. One is to treat it with drugs, and we'll see that's often done in epilepsy. You could treat it surgically. You could use a combination of the two. What do you think was done for this individual? Any guesses? Surgery? No, not surgery, but he had chemotherapy done. The tumor actually regressed. It became smaller. A lot of his personality went back to normal. And the bad part about this is the tumor came back. And when the tumor came back, what do you think happened?
So his personality changes?
Okay, so did he have brand new personality changes, or did he have the same personality changes that we saw here when the tumor was there? What do you think? Same? Different? So he had exactly the same thing. When the tumor started coming back when he was a little bit older, he actually started to exhibit, because the tumor was pushing on the same parts of the circuit, he had actually the same behavior. And so we knew that there was something going on in that circuit that was governing his type of behavior. And these neural circuits, the idea of neural circuits, are very common when you look in neuroscience. It's something, again, I want you to be a little bit more aware of, that in psychiatry, in neurosurgery, neurology, and even in psychology, we are really looking at circuits and how they play a role. You're not really looking at one area of the brain and seeing what happens. You're actually looking at the larger circuit. And this is because these brain regions are structurally connected. These could be neuronal connections or axons. It could have like multiple synaptic contacts. It might be axon A and axon B and axon C before you reach the target. So it might have polysynaptic connections, or it might be functionally connected, where one activity in one region of the brain might be influencing the activity in another region of the brain, and they might be structurally connected there as well. We just don't know about those structural connections yet, or it might be due to other factors. But our basic idea is that brain circuits are responsible, as far as we can tell, for things like movement, affective disorders, anxiety, maybe intelligence, and everything that we can think of in terms of behavior is actually governed by groups of neurons being connected together through these different types of circuits. And we know that when these circuits are working fine, when your circuits between, for example, the VTA and nucleus accumbens and prefrontal cortex are working fine, you're not feeling either anxious or depressed. But if, for example, something in that circuit goes wrong and you have circuit dysfunction, it might lead to a neuropsychiatric disease. Now, I'm going to ask you, because I know you've taken a number of different courses related to neuroanatomy, neuroscience, or psychology. Do we have a model for schizophrenia, which we will come back to later in the course in the second half? So what we're learning in the first half is really sort of the basics that we'll be using again in the second half to examine specific diseases. Do we have a circuit for schizophrenia? Yes?
There's a hippocampal lesion model for
Okay, so there potentially is a hippocampal lesion model for schizophrenia. Do you think there is a circuit involved in epilepsy? Yes, no, maybe? Not sure? Yes.
I think so, because if you, if the people who undergo this, they have really bad seizures, and then you cut the connections between
Okay.
the right hemisphere,
Okay.
it helps.
Okay. So you're talking about a cutting of the corpus callosum so that the seizure activity doesn't spread from one hemisphere to another. Okay, so I'm going to try to convince you by the end of today's lecture that in fact in epilepsy, because that's going to be our model system, that we actually have a lot of connections, that the circuit that's involved in things like epilepsy is actually well developed. And I went through the circuitry of depression with you earlier. As I've covered it in other courses, I'm not going to cover it here, and we're really going to concentrate on epilepsy. I wanted to find something that most of you hadn't maybe studied in a while. And I know many of you have studied Parkinson's disease, some of you have studied depression, and I wanted to cover something that was a little bit more unique. So this gets back to this idea. How do we map out these circuits? So how do we study it? So can we electrically excite them? Maybe, but some of the techniques that we used to have in the 1970s, 1980s, etc., were actually not so great because they had very poor spatial resolution, where we were likely affecting whole groups of neurons at a time, not specific little boxes, but maybe multiple boxes or groups of neurons at a time outside of the region of interest. So you were never sure, am I only stimulating that one group, or am I stimulating multiple groups to see the resulting behavior? Another thing is the drugs that we often use, the pharmacology, oftentimes it takes time to get into the bloodstream, crossing the blood-brain barrier, binding to the receptors. The pharmacology made the timing really poor as well. So we had very poor temporal resolution as well. And of course, once you lesion an animal by traditional methods like chemical lesioning or surgical lesioning, that means that you can't go back, right? Once you've taken a section of the brain out or damaged it, destroyed it completely through chemical lesioning, that area, that box that we had before that's now missing, we can't put it back. And again, we run into the same problem there. How do we know that we've only removed that little box, and how do we know that we haven't removed much more than that? So again, there's some caveats associated with this. How do we know that we're not affecting multiple groups of neurons? How do we know that it's as precise as we think it is? It's only that area in the brain that we are targeting to make sure that it's specific for that group of neurons. So our level of precision for a very, very long period of time, we could not map out that circuit. That circuit in many ways was kind of unfocused. We had an idea. It's like looking at the wall. If you wear glasses, you can make out different patterns, but you can't really see the details. And we needed a way to be able to see the details with a more precise, more refined sort of method. And so one of the big things that different individuals with the molecular biology revolution and genetics in the 1990s and into the early 2000s, one of the things that they wanted to do was, can we get a better way to look at controlling how these neurons are activated and the behavioral consequences of activating these neuronal circuits. Now, many of you will know of this individual here, and I'm hoping that you still remember who he is. Anyone remember who he is? You guys are really bad with faces, right? If you think optogenetics, which is where I'm going, who do you naturally associate with optogenetics?
It starts with a B, isn't it?
Sorry, it starts with
A B.
a B. No, no. So this is Carl Diceroth. You might be thinking, although I doubt it, you might be thinking of the person underneath who had a publication with him as his graduate student in 2005, and this is Ed Boyden. No, we're thinking of Ed Boyden. Okay, and we've all met Fong Zhang. And so Fong Zhang, Ed Boyden, and Carl Diceroth came together as a group at Stanford University. They put their heads together and they thought, okay, we're really good at molecular biology and engineering. We're really good at, he's a psychiatrist, and Ed Boyden is an engineer who came to join his group. How can we actually use our knowledge to go back and fix those issues that we have? How do we get a clearer picture of our neuronal circuits? How do we avoid all the pitfalls of overactivating other groups of neurons? And they came up with a brilliant technique, which is now known as optogenetics. And over the past 10 to 11 years, this optogenetic approach has been used to dissect these neural circuits. They literally have allowed us to go in with precision spatial control to turn on different circuits. So is this circuit really involved in depression? Let's see, let's turn it on using optogenetics. Can we actually go back in and identify a circuit that might be related to things like schizophrenia? So again, maybe we're not 100% sure, but we think we've identified different circuits that might be involved in schizophrenia using optogenetics as well. So this technique has really been a boon to neuroscience. And this technique now is a standard technique in most labs that are dealing with sort of the biological basis of understanding the circuits underlying behavior. Did anyone actually read this paper here, the false memory paper? Have you heard of the false memory paper? Yes?
Is that when they, well, in mice, when they put false memories into mice?
When they put false memories into mice. Correct? Yes? It is more complicated than that. So it's a really dense paper if you ever try reading it. But this is actually really cool. So using optogenetics and using a number of behavioral assays, I won't go into it here because it is actually a really dense paper. It's not by Carl Diceroth's group. The first 3 to 4 years or so of optogenetics, his name was on every single paper related to optogenetics because he was the person to go to. However, in the year 2013, 2014, 2015, a number of other groups started to come out without Carl Diceroth's name. And one of those individuals was Sasumu Tanagawa over at MIT. And he actually showed a really cool experiment last year, in July of last year, very dense if you want to go through and read it. What he did was, and this is technically really challenging, what he did was he identified a group of neurons that were active when a mouse was happy. Okay, like you give the mouse something that it enjoys, groups of neurons light up, he goes back, he tags them, and he uses optogenetics to turn those neurons back on. And then he makes those mice depressed because he knows the circuitry of depression. And he wants to know if those happy memories are there, is the mouse still going to feel as depressed? And what he's able to show, at least in a mouse model, using optogenetics, which reveals the power of optogenetics as well as the underlying idea of the circuitry of optogenetics, is that if you activate those happy memory engrams that existed before, that the mouse isn't quite as depressed as it was before. Now, in humans, that might be much more difficult. Like if you're depressed already, thinking of happy times may help you, but it's really hard to engage in those happy thoughts when you're feeling depressed to begin with. But we're going to actually concentrate not on any of these, which are classic, but we're going to concentrate on epilepsy. And I'm going to take you through a couple of things why I think this model of epilepsy and optogenetics is a really cool one. Because optogenetics is no longer just pie in the sky. It's actually being done in humans. And my guess is the next study that will be done, probably in humans, will actually be done in patients with epilepsy. And hopefully by the end of this, you'll understand the reasons why. So the basis of his technique is really, really simple. And by the way, I don't want to give short shrift. He won a $3 million prize this year in I think it was 2012, where the pot wasn't quite as big. He won a $2 million prize with the individuals up here. And one of those individuals, George Nadel, who you may or may not know, who's right above Carl Deisseroth up here at the very top, actually he was the first person to publish a paper in 2003 that Carl Deisseroth read on optogenetically engineered proteins in the in in a C. elegans model. So he read this paper that George Nadel had published, citing that these particular types of algae that have a certain color associated with them, this Chlamydomonas reinhardtii, when he identified the opsin protein that was activatable by light, he thought, wow, this might work, because your brain is in an area that's normally not exposed to light. When this opens, a channel opens, the neurons have channels to begin with. Why don't we use this to our advantage? Shine light in a dark place, open up channels which normally happen in neurons anyways. And these things open up on the order of several milliseconds, which is just like the neurons do. So it might work to activate neurons just like their native state. So using transgenic methods, Carl Deisseroth's group in 2005 through 2008 started experimenting. Could we actually put this in C. elegans? Could we then modify C. elegans behavior? But his big claim to fame is actually being able to put it into mice as well as into rats. And when they did that through stereotactic viral injection, they were able to show that you could indeed change a circuit, firing of a circuit, and then change a behavior in an intact animal. And this specificity that had never been shown before was something that was really, really important. As soon as people read that paper, everyone realized this is something incredibly powerful, and everyone should start using these different types of techniques. And so the paper I'm going to concentrate a little bit more on, and you can see Carl Deisseroth's name is on this particular paper. This is an earlier paper back in 2009. This paper here is the one that I'm going to concentrate on, and this is sort of an on-demand optogenetic control. And what this means is eventually we're going to understand epilepsy, we're going to understand animal models of epilepsy, and then we're going to go back and revisit optogenetics as a control for epilepsy. But what this says is if you're having a seizure, you don't have to take a medication. If you have optogenetics in an animal model, as soon as seizure activity starts, optogenetics quiets that on demand the activity of those neurons. So seizure activity down the road may be something that is controlled at will through optogenetics. And we'll go through that paper in more detail on Thursday. It's an amazing paper from an engineering standpoint, a behavioral standpoint, and what it might actually mean to people who have epilepsy. It is likely in a class of about 70 students that 1 or 2 of you will know individuals that have had epilepsy. And we're not talking about seizures, and hopefully by the end of today, you'll know what the difference is between the two. So when they went back and looked at the different types of algae that were expressing different types of opsins, and these are all examples of opsins. These are proteins that act as channels, and these channels open in response to light. And that's what an opsin does. It's like the rhodopsin in your eye. Your rhodopsin responds to flashes of light. So when this happens, and this is the classic one of the things that they isolated from that clitomonas was this CHR2, the channel rhodopsin, the granddaddy of all of the optogenetic methods. And when you shine blue light, if it happens to be on a neuron, it will allow the passage of both sodium as well as calcium. And for all of you who have taken any physiology courses, when a channel opens and sodium and calcium flow down its concentration gradient, what happens to the neuron that's expressing this channel? So it depolarizes, and potentially, if it gets to enough of an excited state, it can actually start firing action potentials. Later on, they found that there were other types of algal blooms that express different types of opsins, and these were actually found in the Sahara Desert, and there's a lake there that actually have different types of algae that grow there, and they sort of fluoresce a reddish color. They kind of look more reddish when they're in the water. And when these get stimulated either by red or orange wavelengths of light, they will actually cause the influx of either chloride or the efflux, hydrogen, out of the cell when they are responding to light. And again, these are the halorhodopsins, and the halorhodopsins respond in the opposite way. So what happens when negative charge enters the cell? That cell becomes hyperpolarized or less likely to fire action potentials. And when positive charge leaves the cell, what happens to that neuron or cell? So it also becomes hyperpolarized as well. So the beauty of this is whether you have the channel rhodopsin that excites the cell or the halorhodopsin that inhibits the cell, they respond very, very quickly. And what is being shown down here is that when you have a little flash of blue light, when you have that blue light on, you will be able to trigger action potential generation in this particular case. And you can see that if the same neuron is also expressing the halorhodopsin, the ones that normally cause inhibition, that when you trigger excitation, that little blue flash of light, this is the action potential going up, it's going closer to the threshold value. When it triggers this, in the presence of this inhibiting light, you will actually prevent excitation from occurring. So it works remarkably well. You guys believe this is true? Yes? Yeah, pretty believable. You've seen this multiple times, right? Tiangi, does this actually work? Yes? Yes? Okay. Actually, not as well as it says here, though, right? Because one of the issues is this transference of chloride and hydrogen isn't as effective as the movement of sodium and calcium. And so the next generation of these halorhodopsins is really good, but the first generation actually turned out to be kind of iffy. And we'll talk about that in the next lecture. And I have to apologize. I don't think that this is going to actually play. It just shows you how action potentials get generated down the axon when these channels are there. We have to introduce them. They're not normally found on these neurons, and these are something that we have to be able to introduce on them, and we have to provide it with light, okay? So I also wanted to introduce you to sort of the next generation of optogenetic tools. I told you now, in the United States, there is a clinical trial for using this system in individuals that have chronic pain. If you're in a lot of pain all the time, and there's no effective drug that works to turn those off, individuals sign up for these different types of clinical trials where different types of channels are going to inhibit activity in pain pathways. But these are sort of the next generation of optogenetic tools. Some of you will have heard of chemocogenetics or pharmacogenetics, where you use a drug to activate either the DREAD system or others. But there is sort of another version in optogenetics where using light, we can actually stimulate G protein coupled receptors, and those G protein coupled receptors can activate the GQ pathway or the GS stimulatory pathway to increase cyclic AMP inside the neurons with a flash of light, or decrease the level of cyclic AMP with a flash of light as well. And the beauty of this is that many of the systems that we study aren't sort of on off right away. They take time, and they need these metabotropic effects to accumulate. And then this last group, which is really exciting, if we can introduce these inside the cell, are these bacterial cyclases, which can also be stimulated with flashes of light, which might allow for more effective ATP exchange, as well as the formation of cyclic AMP inside the cell. So it's a metabotropic pathway that we're stimulating here with a flash of light inside the cell. Okay, and if you're wondering, how does the light get inside the cell? Remember, your cells are transparent, right? Your neurons actually aren't color-coded. If you can get it inside the cell, you'll be able to stimulate this bacterial cyclase. Now, this is a really busy slide. What I want you to take away from all of this, because the Opto-XR is actually on the previous diagram, is that this next generation that has already been published involves what is known as the SSFO, or the step function opsins, which have been stabilized. So before, when you had to stimulate for long periods of time, you had to literally have that light on for long periods of time. And what these SSFOs do is, once you flash on that light, these opsins stay active for long periods of time, in some cases up to 30 minutes. So once you activate a cascade, you don't have to keep flashing that light on and off, which might be bad for the neuron. You flash it on once, this opsin stays on for up to 30 minutes in a non-metabotropic way. The other thing that makes this really unique, and this is the second point that I want you to take from this slide, this is sort of the next generation of optogenetics, is in theory now this next generation of stabilized step function opsins that allow for longer depolarizations are also much more sensitive. You don't actually need an optical fiber cable anymore. You can actually do this from the surface of a brain. You don't have to implant an electrode. I think most of us would actually feel really weird having an optrode implanted into your brain, but now you can stimulate directly from the top of your brain. And this right now can penetrate down to about 3 millimeters or so. So that means that before, we were really limited to small animals like mice or rats. In theory now, at least in theory, because it's less invasive, we don't have to put in a fiber optic cable, we can actually stimulate directly on the surface of the brain. It can shine light through. This step function opsin that's been stabilized can be activated, and larger brain regions or larger brained animals can now be studied as well. So it's really exciting. So as this continues to progress, and those last 3 points about the value of these step function opsins and the stabilized step function opsins lasting longer, that's point 1 that you need to make sure that you are aware of. And then 2, that we no longer need a fiber optic guide. You can sit on the cell, on the brain surface, and this now allows for larger brain regions to be activated, as well as larger brained animals to be investigated as well. So those are some exciting things about the next generation of optogenetics that have already been published and are already out in the literature. So this is not a trivial technique. It requires a number of different steps that you need to be aware of, and you need to know them because you need to be able to evaluate. Is this something that actually is done the right way, or is this something that is maybe at fault? So there are a lot of different things, and these are sort of the generalized 6 steps. There are a lot more than this, but if you break it down, we need to have a construct that actually contains the light-sensitive ion channel, something that has an opsin in it, a channel rhodopsin, a step function opsin that's been stabilized, whatever it is, we have to engineer this ourselves. Then we have to package it into something that can deliver that payload to neurons. And as we learned previously, viruses are both myotropic, but viruses are tropic. They like infecting neurons as well. We have to get that into the brain, and then we have to have a way to stimulate the different opsins once they're expressed by putting in a fiber optic cable. And again, the next generation, you probably don't need it. What they are suggesting that will happen is you will have a very, very thin light emitting diode that's placed on top of the brain and then shines light whenever it needs to shine down on. You don't have to penetrate the brain at all. But in this case, you need an an optrode or a fiber optic cable going in, and then you need to stimulate it at specific frequencies, and then you need some kind of behavioral assay to actually see, is this working, and is this whole circuit that we've interrogated using this actually the circuit that we thought was going to be at work. So these are the three different methods that we are going to see, and in fact the first two are the ones that we're going to concentrate on for the first little while. And many of you will have already had the different versions of this or different slides related to this. So we have to stereotactically inject to an area that we know or are interested in. Remember, we're looking at circuits, those boxes. We think those boxes are connected by these different connections. We're not 100% sure, but we would inject into like one group or one box of these neurons, and then see what happens when we have this group of neurons being expressing these opsins. When we stimulate these opsins and they send out projections to both A and C as part of its normal circuit, what do we expect to observe? And does our prediction actually hold true? We could also make sure that it's only affecting this particular group of neurons, and we're going to talk about Cre recombinase in a few moments. And we won't talk in this course about projection type of optogenetic experiments. And for the time being, I'm going to focus on the top two, the one on panel A as well as on panel B. Okay, and I think now is actually a good time to take a break. I only have about 12 more slides, but I do want to go through them fairly slowly, and I do want to give you some time to get into groups today as well. Again, you don't have to worry about memorizing the details of this slide. I'm going to tell you what you need to know from today's lecture as we go along. So why don't we take our break now? We'll come back at 10 after. They'll take you through more about optogenetics and Cre recombinase. Then I'll take you through temporal lobe epilepsy to set up the paper for Thursday. To get going. Up until a few years ago, before the creation of optogenetic Cre driver lines, which I'm not going to get into any detail, one of the things that you had to do in order to increase the efficiency, increase the precision of optogenetics, so that if you're targeting only group A neurons, that any of the other nearby neurons are not going to be infected, you can do lots of tricks like using specific promoter sequences. But another way that you can do this is by generating a Cre lock system. And the paper that I'm going to present to you on Thursday actually makes use of a Cre lock system in the cholinergic system. And so I'm going to need to revisit it. So beforehand, I wanted to go back, if you haven't had this before, to talk about specificity through the Cre locks system. So this is one of the ways in which in genetics and molecular biology, we increase the precision and specificity of gene delivery as well as gene expression. And so there are a couple of really key points and key components to this. So if it's been a while since you've had this, or you might have forgotten, this Cre lock system actually doesn't exist in eukaryotic cells. It is not normally found in mammalian cells. It's not found in the mouse, not found in the rat, not found in you. And this Cre lock system is a way that we can engineer things because it's normally found within bacteria. So this Cre lock system that we generate allows us to insert sequences, which we call these LOX P sites. And these are 34 base pair sequences. We artificially generate them. We are creating this sort of step one. We are putting together this genetic construct, and inside this genetic construct, one of the things that we will generate are these LOX P sites. Now, this gene X is likely going to be one of the channel rhodopsins or a halo rhodopsin. But in order to make that halo or channel rhodopsin, this gene X, work more efficiently and more precisely, we want to make sure we have some Cre lock sites around it if we need this to be existent. So these LOX P sites, these 34 base pair sequences that we artificially generate around the gene that we are putting into this construct that we're eventually going to package into a virus, is really unique in the sense that Cre recombinase, which is a protein also not normally found in eukaryotic systems, binds specifically to two of these LOX P sequences. And how it binds and as a recombinase, how it cuts as an enzyme is really important. Because if the LOX-P sites are in the same direction, then one of the things that will happen is when the Cre recombinase binds to these two sites that are in the same direction, 5' to 3', one of the things that happens is the gene gets removed. Whatever's in between the LOX-P sites that we've had now gets removed. The only thing that's remaining in our construct, the gene construct, is the LOX-P site. The other way that we can do this is not to put them in the same orientation as we see up here, but is to invert the sequences to make them in the opposite direction as you see down here. So if we take the LOX-P sites and don't put them in the 5' to 3', we put this one in the 5' to 3', and we put this gene in between, and then we invert, or we use a backward sequence, however you feel comfortable talking about it, and here I've mentioned that it's in the opposite orientation, then when the Cre recombinase, because Cre recombinase always binds to these two sites, when it now binds here, and then it tries to excise whatever is in there, it can't do it. But what it will do is it will actually invert or flip the sequence that's found between these LOX-P sites. And you need to be aware of this. And now you might be thinking, well, removing a gene kind of makes sense. But you're also going to see that doing this lower panel actually makes a lot of sense as well. Okay, and we'll come back to that a little bit more on Thursday. Do you have any questions about this? Of course, Danny. What can I help you with?
How do they ensure that it doesn't flip again?
Ah,
Because LOX-P remains,
yes.
both LOX-P are there, so why doesn't it flip more?
Yeah, so actually, that's a great question. So why doesn't it just keep flipping back and forth over and over again? One of the things that you can engineer that isn't on this diagram, and I'm not sure it's on the diagram for Thursday either, you can actually generate another type of LOX site over here to actually prevent that once it's flipped, it can't flip again. So there are techniques that we use to generate these non-invertible sequences after the first inversion. Yes?
Why does it split? Like, what's the logic behind it?
So the Cre recombinase is trying to cut the strand of DNA. It will try to bind it and it will try to excise it. But when it does so, it actually just ends up inverting it rather than being able to excise both ends. Okay. Any other questions before we go on? So again, this is a really powerful technique, and I come back and introduce it to you again on Thursday in a specific context related to epilepsy. I wanted you to be at least aware of the different strategies that are used to increase the specificity. So the second step was to actually be able to deliver this. So imagine we've just engineered this thing. You've sat there as part of your summer research project. You were given a template. You were asked to use PCR to insert these 34 base pair sequences. You were asked to clone channel rhodopsin. Everything worked. The sequencing worked. You then package it into a virus that allows you to take that genetic construct that you've created and deliver it to a neuron. Because naked plasmids, if I just took the plasmid by itself and I inserted it into the brain, it doesn't work very well. Genetic material doesn't get taken up very easily by your neurons, at least in the brain. And even in culture or monolayers, it doesn't work very well either. So we need to be able to deliver it inside the cell, which is where we want it to go. And one of the ways that we know genetic material can be delivered really well is that we can use a virus to package the genetic construct that we have engineered ourselves and to deliver it specifically to a group of neurons. Often the type of virus that we use is an adenovirus or an adeno-associated virus, which is the AAV that we've encountered a number of times. We don't want the virus to keep replicating, and the AAV or the adeno-associated virus is a replication deficient virus. Whatever you inject in there, in theory, is going to stop dividing. It'll just deliver its payload. So I usually ask this to my neuroscience students, so maybe I can ask the genetic students. Can you give me any real-world examples of how you know that viruses can infect neurons and why it's such an effective system? Yes.
Rabies?
Rabies? Okay. How does that work?
replicates and goes to part of your brain and makes you like angry and stuff, and then it also goes to your saliva glands.
Okay, so it travels along the axons near the place where you've been bit, and it goes into your central nervous system. Absolutely. Anything else? Yes.
Shingles and sometimes herpes are known to live in the nerves, right?
That's correct, and many of these, and one of the reasons why I'm really interested in neuroimmunology is for precisely that reason. These viruses tend to hide out in the nerves. Your immune system isn't as efficient in attacking neurons, and so if your virus is there hiding inside a neuron with things like shingles, or the chickenpox virus, then we know that they can live for years at a time until they're ready to start replicating. You guys also remember this slide, right? Yes? So this is an important slide for you to know. I'm going to be very honest with you. I'm not going to ask you a short answer question related to coming up with this chart, but I'm going to ask you to demonstrate that you understand this chart through the use of a short answer question, but you don't have to memorize this chart for the sake of replicating this chart. So there are a couple of things that I'm going to point out to you. Some of you asked this in the last lecture. So why do we use different types of viruses? What's the inherent value or advantage to using, for example, an adeno-associated virus, whether it's type 5 or type 9 or whatever? And the reason is for the adeno-associated virus, one of the beautiful things about this is that we can use DNA, and we can use single-stranded DNA, which is a PCR reaction. So we can generate these things really, really easily. The problem is there's not a lot that we can pack into an adenovirus. It has limited capacity for genetic material. We can only put in about 5 kilobases. Remember Fong Zhang's issue, like delivering both Cas9 and his guide RNAs and the promoter regions? So this is the size limitation that we have with adeno-associated viruses. The speed of expression. So you inject in the virus, you put it into a group of neurons. How long do you have to wait before you can start flashing blue light? You have to wait a few weeks. It takes time for this to come up in terms of expression. But once it's there, those groups of neurons, in theory, and people have gone back to check behaviorally, they stay there for a very long period of time, at least on the order of years. So another very common type of neuronal system that you'll read about, the group of papers that I'm going to share with you do not use the lentivirus, but other papers do. The lentivirus is unique in the sense that you can generate, you need the genetic material to be coded as RNA. So it's an extra step for a molecular biologist to generate these template strand DNA, convert it to RNA, then package it into a lentivirus. We know that the packaging, it's a little bit bigger, about 8 kilobases, not much. 3 kilobases is actually not that much bigger in terms of the genetic material that you can pack in. Again, very similar in terms of speed of expression. So the herpes simplex virus is actually also kind of neat. It uses DNA as well. We can package much more into this. The speed of onset, as soon as you infect neurons, you can start seeing the expression of GFP, for example, a non-endogenous gene that's being expressed on the order of a couple of days. You can see that very clearly in the brain after infection. It doesn't last as long though, so it tends to disappear with time. But there are some molecular techniques that you can use to make sure that it lasts a little bit longer. But those are sort of the advantages and disadvantages of why we choose to use some vectors and not others. Do you have any questions about this before we go on? Danny? No? Okay. So I'm going to give you a little bit of background, and I'm going to get you to think as sort of geneticists as well as neurobiologists. And we are going to come back in terms of pure genetics later on in the course related to epilepsy. It is actually a very interesting disorder to take a look at. So some of you have mentioned seizures and epilepsy. They are not the same thing. And if nothing else from the end of today's lecture, you should be clear on what epilepsy is versus what a seizure is. It is likely, or sorry, I shouldn't say likely, it is possible that you have had a seizure once, but you don't have epilepsy. You might go drinking on a Friday night. You might get really intoxicated. You might seize, but you don't have epilepsy. You may never seize again for the rest of your life. So epilepsy and seizures are two things that we're going to talk a little bit about. So epilepsy is a recurrent spontaneous seizure activity. There are a number of things that can actually cause this. There are a number of disease entities. They look very different. You can have myoclonic tonic seizures where your muscles are actually contracting very severely, and the myoclonic tonic type are very different looking than absence type seizures. So they can have different phenotypes. The mechanisms are also very different overall. Some might involve calcium channels, some might involve GABA receptors, some might involve changes in glutamate receptors. The brain structures that are targeted, as well as the overall prognosis, might be very different. Epilepsy is not a uniform disorder. Okay, so if you say that you have epilepsy, it's just like saying I like food, right? And it can mean a lot of different things to a lot of different people. Can you think of any disease states or disease entities, as written here, that might cause epilepsy? So you don't have a genetic predisposition. What might cause you to have epilepsy? Another disease that might cause you to have epilepsy. Yep.
Isn't it kind of related to like overactivate or like overexcitation?
Yeah, it's completely possible because it's a circuit, and this involves a dysregulation of a circuit. So what diseases do you know that involve circuits that might involve this circuit? Any ideas? So neurodegenerative diseases often lead to the formation of epilepsy. So there are a number of those. We won't talk about all of them. We'll mention them in passing on Thursday. But clinical diseases can also be acute. You could have head trauma, and that might lead to epilepsy as well in some individuals. So if you have epilepsy, you have seized more than once, you've had a seizure more than once in your life, and this can actually affect your life dramatically, depending on the type of seizure activity that you exhibit. So a seizure is any kind of sudden change in sensation. It doesn't mean that you're like on the floor with spasms. It can mean that a change in sensation or awareness. It's usually due to an electrical disturbance in your brain that we can measure on an EEG or through other mechanisms. And one of the things that happens is that your brain is normally firing all the time, and your brain is firing in an unsynchronized way all the time. Your neurons are kind of all doing their own thing. But if, for example, suddenly all your neurons decided at the same moment to all do the same thing at the same time, that is an example of synchronization. So imagine this. Imagine that it is the middle of the day here at U of T, and every last building that's full of students decides at the same time that they're all going to let out at the same time. And you're trying to walk from here all the way up to Harvard Street. It's going to be really tough because everything is synchronized. You're going to have a roadblock, which is what sort of happens during epilepsy. So everyone has a certain seizure threshold. I won't get into all of the details about this. There might be a genetic component to this as well. For whatever reasons, for some individuals, that seizure threshold might be lower than it is for other individuals. If you have more than one seizure, if you have more than two seizures, then you clinically have had or you have epilepsy. Okay, so if you have one seizure, it's fine. It can happen for a number of reasons, but it is when you've had more than two seizures that you are clinically diagnosed as having epilepsy. So epilepsies can be categorized into two big types. Can either be generalized epilepsy, or you can have a partial focal type of epilepsy as well. One of the ones that you are probably the most aware of, or you've heard about the most often in literature, is what's known as an absence form of epilepsy, or a petit mal type of epilepsy. And one of the things that happens here, they're not rolling on the floor, they're not convulsing, they don't have that myoclonic tonic. You might be sitting beside someone and they might have this petit mal seizure activity. You wouldn't be aware of it. They just lose consciousness, right? Is that a big deal? I do that all the time in lecture. In fact, I had just done that in lecture until you reminded me to come back. It is a big deal. Imagine you are in grade 2, and in grade 2 you're having these absence type of seizures in this form of epilepsy, and you're learning the multiplication table. And you look up, and they had just told you what 2 times 2 equals, and then you look up again, and they're just going through the 3 times table. So you're behind, and you're going to be behind because you've been blanking out. You've had this change in your level of consciousness where you're not aware of your surroundings, and it has a dramatic impact on your everything. Your personality, the way that you interact, your social skills, your cognitive skills, all of this. So it may not sound like that really bad form of epilepsy that everyone characterizes in the movies and on TV where someone's on the floor convulsing and seizing. Certainly that happens, but this is just as bad. Okay, this loss of awareness and consciousness. And we know that we can actually measure this really strongly on an EEG. And one of the things, again, because it's generalized, we see this on widespread areas of the brain simultaneously. Does that make sense to everyone? Yes? Oh, but that's a bad thing for you to agree with. Sorry, you missed your chance. Okay, never mind. Yeah. No. Let's not be Canadian about this. Let's go on. Yeah.
The question was, like, I guess I don't know if it's like common, just if not actually the way it is, but it's what I learned that certain things, if they're spontaneous and having certain things, like I guess it predispose you to having seizures more likely, or like loud music, or like flashing lights.
Possible. Yep.
Is that just a common? Is that just because overstimulation of the brain could potentially?
Yep. Possibly. Yes. That does happen, Danny. That's actually true. There are dogs, for example, that are inbred, that when they get strobes of light on them, they actually will also have seizures. Yeah. And dogs are actually it's very common to have epilepsy in dogs as well. If you're thinking about this, I want you to go back to what I told you earlier, right? And I want you to be really good neurobiologists and or geneticists. Is that actually correct? Every area of your brain, if it's in a part of a circuit, every area of your brain simultaneously is going to be active. That happens? Yes? You guys are in neuro. Tell me how that happens. Yes.
But like, wouldn't, for that to be able to happen, wouldn't your GABAergic neurons have to like switch so that, because they're inhibitory?
Sure.
So,
But it happens everywhere in your brain all at the same time. Like the neuron over here says, oh, by the way, I'm about to turn my GABA off, and it somehow magically tells that to the other part of the brain, because widespread areas of the brain all at once are going to shut off. Everyone gets out of school at the same time. How does everyone tell them? Think like neurobiologists. Think about what you know about neuroanatomy. Think about how the brain is a circuit. What is the driver in the circuit? Is there a driver in the circuit? Is there one thing that kind of links up everything? Sorry? Are you a neuroscientist? Pretty good. I like that. So if you go back to what we learn in physiology and anatomy, the thalamus actually links up to many different regions of the brain. It's possible for your thalamus to signal everywhere spontaneously because it sends out thalamocortical projections that every area in the brain starts to act in the same way at the same time. So in terms of seizures, again, please don't bother memorizing the numbers. It's more to give you an idea of the scale and the scope of what is going on. The incidence of this is about 80 out of 100,000 individuals per year. The lifetime incidence over the course of a lifetime, about 9% of individuals will have some sort of seizure activity. And in this particular case, about a third of these are induced by fevers. So as Danny was mentioning, there are some stimuli that cause you to have seizure activity. In some cases, especially in young individuals, having a fever or fever-related can lead to a form of seizure activity that causes them to have these convulsions or the tonic clonic type. Epilepsy, though, where you've had more than 2 seizures, has a lower incidence. about 45, not quite half of what it is for seizures, but about 45 out of 100,000 individuals per year. And the cumulative lifetime incidence of this is much lower as well. About 3% of individuals will have it at some point in their life if they are predisposed to having epilepsy. So it's not a trivial, it's not a rare genetic, or it's not a rare disorder. Some of it is genetic, not all of it, but it is actually more common than we give it credence for. So there are lots of different things that cause seizure. You might have had an injury during your prenatal term or during birth. It could be because of an inborn error of metabolism. I don't have this slide, unfortunately, anymore. I took it out of the slide deck, but we know that, for example, your levels of GABA and glutamate at a very early age is actually really important as to whether or not you will develop seizure activity or not, and that's because of your early metabolism. It could be that an area of your brain has a congenital malformation. If it's a circuit issue, having a malformation in part of your circuit that is identifiable may also cause seizure activity. If you have childhood forms or adolescent forms of seizures, oftentimes this is either of unknown origin, idiopathic, it could be genetic. Some forms of seizures and epilepsy come on during childhood, then disappear. Some come on during childhood and adolescence, then disappear, and some don't come on until adulthood. And there are lots of reasons for that, which we'll come back to when we talk about the genetics of epilepsy. You can also develop seizures, again, for a number of different clinical manifestations. One of those is CNS infection. So if you have an infection in your CNS, bacterial or viral, you can also induce seizure activity. You could have a really bad blow to the head that may induce seizure activity as well. We know that we can detect seizure activity and epilepsy using EEG, this electroencephalogram, where we're using electrodes on the surface of your skull to measure the electrical field that's being generated. And many of you have done this either in psychology or in your neuroscience labs. Okay, and again, we're kind of looking at a whole group of neurons, millions of neurons that are being synchronously active to allow us to record on the surface of the scalp. And when you guys do these types of EEGs, do you guys use this many electrodes? Probably not. Do you guys use the bathing cap? Yeah. So this is actually more of a standard clinical type of EEG. It's actually not much more difficult than using like 2 or 3 lead EEGs. You just need to map out where they're going to go. Here's an example of the types of things that I was mentioning to you earlier. This individual using these different leads all over the surface of their skull. Each one of these little squiggles represents one electrode. So here's one electrode, here's another, all the way up to the top. Again, you can have up to 256 if you really wish to have a dense electrogram. But you can see that here it's nice and normal, here it's nice and normal, and it stays nice and normal until something happens here. And here, everything goes on at the same time, everything. Right? So this is an example of where everything decides at the same time without it talking to each other, because you have electrodes at this part of your brain and you have electrodes back here, and here. They're all doing the same thing at exactly the same moment in time. And so this is an example of a generalized seizure. It is taking place everywhere in the brain at the same time. I told you one example of a generalized type of seizure is this absence seizure, which is a petit mal type seizure. And again, for most individuals, it sort of starts off with an impairment of awareness. Like, I kind of zoned out, like not the usual zoning out because you're tired or your prof happens to be talking in a monotone and you're kind of going in and out, but you just literally lose consciousness. You're not aware that you've lost consciousness. You're not closing your eyes. The person sitting beside you may not even be aware that you've lost consciousness. And this can last anywhere from a few seconds to 20 seconds. And again, imagine like losing 20 seconds of your life at like any time of the day. Not something that is pleasant for the individual. Again, sudden onset, and it also disappears, sudden resolution. The onset of this, though, is typically early in development, and usually, not not always, but usually if you have these types of absence seizures or petit mal type of seizures or epilepsy, these resolve by the time you're 18. And again, for epilepsy, it's this really unique disorder, some of it genetic, some of it environmental, etc., where it really goes back to the circuit. And when things develop in that circuit and when certain switches are turned on in this circuit, and here it actually happens to turn off by the time you reach adulthood. Tell me what's going on here. So this again, separate leads. So here's 1, here's 2, here's 3, slightly different. What do you think is going on here? Arslan?
Focal seizure or motion seizure?
So tell me where you think it's starting or which lead we're picking it up on first.
It starts when it gets the amplitude increases very rapidly.
So right around this area, these 3 leads, we start to see increased activity. So here's nice normal baseline that we're picking up on our EEGs, and suddenly it starts off here in these 3 leads, and then it spreads throughout the rest of the brain. And this is an example of a focal seizure that spreads, and we can again detect all of this through EEG. EEG, a lot of you think that, wow, you know what, we need to use these really fancy fMRI machines, or we need to use MEG devices. They're great, but EEGs are enough to pick up synchronized activity really easily in an individual, just like you're seeing up here. So some of you were mentioning this is maybe due to GABA, and certainly we'll see that GABA plays a big role in this. So when you see the brain doing all of this that you see in this particular slide, do you think the brain is really, really quiet at this point in time? It's going off like crazy. So I told you in your brain, for example, you have glutamate receptors and GABA receptors, glutamate being excitatory, GABA being inhibitory. So there are really 2 choices. One, the glutamate system is being overactive, or the GABAergic system is being underactive, right? So we're stopping inhibition or decreasing inhibition. That means excitation is going up. We've taken our foot off the brake, and that allows us to speed up. Or we put our foot on the gas pedal, which is what increasing glutamate receptors would do, and it all speeds up. But it turns out that for the most part, the drugs that we look at that treat epilepsy are either targeting calcium channel receptors or they're targeting GABAergic systems. And part of the reason we think that GABA is really essential in this whole process is that if we look at these GABA receptors, which you see down here, and you should know the structure of the GABA receptor, and you should make sure that you understand why GABA receptors might be important in different forms of seizure activity and epilepsy. If we look at the overall structure of the GABA receptor, we see a number of different sites. So ethanol binds to GABA A receptors. We know that benzodiazepines, which make you feel nice and mellow, nice and relaxed, also bind to GABA A receptors. Barbiturates also bind to GABA A receptors. And when all of these plus GABA bind, one of the things that happens is that chloride flows in and it quiets the activity of whatever neuron this GABA A receptor is expressed on. And one of the things that we think happens is, for whatever reasons, your brain becomes much more excitable in epilepsy during a seizure. And one of the things that we think is a normal break is this GABA. So how do we increase the GABA inhibition in a brain that is going like crazy, where you have the foot on the gas? How do we make sure that we're also starting to put our foot on the brake, which is represented by this GABA A receptor? Well, we can't increase the number of GABA A receptors easily, but we can increase the availability of GABA presynaptically. So we can increase the amount of GABA, hoping to stimulate more and more of these GABA A receptors. And one of the drugs that is targeted is known as tiagabine. And tiagabine, among others, actually acts to inhibit the activity of this GABA transporter called GAT1 presynaptically. Just like in depression, we can prevent the reuptake. Here we're preventing the reuptake of GABA, which means that there's more GABA available out here in the synaptic cleft, more to bind here. And so the neurons might be going crazy, but here is a way to break or inhibit the overactive neurons, potentially using tiagabine as a drug. But again, think about this, right? You've just had, for whatever reasons, an overactive seizing brain. You're going to have to take tiagabine, wait for it to start working, hope that it's going to cause the excitation to die down. For someone who has like different forms of epilepsy, whether they're seizing and convulsing, or whether they have absence seizures where they're not sure it's going to actually happen until it happens and they're not aware, it's not a very appealing sort of treatment that's available for these individuals. So in particular, sorry? So in particular, where we have things in these partial or focal epilepsies, to start off in one region of the brain. And this is what, again, all of you in neuroscience and in psychology are aware of in terms of temporal lobe epilepsy. We start off in a small area being activated. So that previous EEG that I showed you before, and then it spreads. Then it gets to other parts of the brain through those circuits that I mentioned to you earlier. I mean, the hippocampus, as part of the temporal lobe, has circuits and connections with other parts of the brain, and it will then involve the entire brain, but it starts off in one area. So imagine you know that. You know that because our technology is really good. We can actually go in and map out very, very precisely the region of the brains, the region in your temporal lobe that shows increased activity. We can do it through EEG, we can do it through MEG, whatever it is, we can actually map out spatially what areas of your temporal lobe is going to be active and then spread to other areas. A lot of you will remember HM, and HM had this really bad form of epilepsy that actually impeded his lifestyle. He had it surgically removed, right? He had his hippocampus removed from the temporal lobe, and this temporal lobe epilepsy still is treated surgically. People still get parts of the temporal lobe removed. They get it treated through electrical means as well, and we'll talk about that in the next lecture. Overall, the drugs that we have take time to work. The other options like surgery or electroshock are not very appetizing. And so if you have epilepsy, which is not that uncommon, and one of the things I hope you've taken away, and you're looking at a treatment that will help make your life a little bit less uncomfortable, then imagine being able to, as soon as an epileptic seizure, the synchronization starting to occur, it's detected, and then you have a way to turn it off. And this is sort of the beauty of what optogenetics allows, at least in an animal model. And I'm going to try to convince you on Thursday that this system that they've engineered, where they have introduced optogenetic probes into an area of the brain that gets overexcited, that as soon as that excitation through EEGs gets detected, a light flashes on and it inhibits the activity in that area of the brain. Do you think that would be attractive? I think it would be pretty cool if that type of clinical intervention was available in humans. So let me ask you, just so that I'm aware how much you've taken away from this. If you went back to the slides that I told you earlier, and then I'll give you a few minutes to get into groups or talk about what you need to talk about, that system that I mentioned to you earlier, where that paper that I'll be talking to you about in Nature Communications, that system looks at rising excitation, it detects this rising excitation, and then as soon as it does that, it triggers an automatic switch that the animal isn't even aware of, and it flashes a light. What system would you use to be able to turn it back down? Would you use a channel rhodopsin blue light system? Would you use a halo rhodopsin orange light system? What would you use? Yep. So you probably want to use halo rhodopsins, reduce activity in the areas that is spontaneously becoming more active. And we'll see if Oriana is right on Thursday. Do you have any questions about this before we sort of wrap things up? Yes.
Just wondering, what are the drugs like the tiagabine?
Yes.
When do they want to take them prophylactically? Like, do they cause side effects?
Yes. Yes, they do. So these drugs like tiagabine, for example, cause side effects. The calcium channel blockers that are also available for individuals that have epilepsy cause side effects. So you don't want to be taking them all the time. By the way, individuals that have like certain forms of epilepsy, they take it prophylactically. The issue is, imagine you're a teenager, right? Do you want to take drugs all the time?
Well,
Well, I asked that in the wrong way. Do you want to take medication all the time? You probably forget, right? And you probably, just like my kid forgets to clean her room every time I ask her to, you probably don't want to be on these medications. And that becomes an issue because it's just like, oh yeah, you know what, I'm okay. Like I'm gonna study like next weekend, I'll be okay. And then before you know it, the exam hits you. It's the same thing. People are on these for a while. They get great results, no seizures for a while. I'm okay. They stop taking it, and then they seize. Right? They might seize at an inopportune moment, and that's one of the things that sort of happens. Some individuals do take it prophylactically, but again, there are side effects associated with it. Not everyone adheres to that prophylactic regimen. There are a number of issues that are associated with that. It is a great question, though. Any other questions? Okay, so we're going to take a look at sort of the paper that comes along with this as sort of a genetic technique that we think has therapeutic implications. It will have therapeutic implications in humans. I'm going to stop here a little bit early to ask you to make sure that you know which groups you're in. Again, just to be really clear on this, I haven't posted any of the assignments yet. I will over the next week or so to give you some more guidelines. Your first assignment as a group, and you don't have to stay together in that particular group, and I don't actually mind if that first group that you're in is a little bit bigger than 4 members. 5 or 6 is fine too. What I'm going to ask you to do is, as a group, rewrite the midterm, get a better mark. Hopefully, you can't do any worse, but get a better mark on the midterm. That will be your first group assignment. And then secondly, you will do, you'll pick a paper as a group, present it as a group, and you'll get guidelines on that, and then do a grant proposal as a group as well that your TAs will mark, and all of you will get the same mark on that. We will have workshops related to doing each of those in turn to make sure that you know what the expectations are, but you don't have any guidelines right now, which is what one of your colleagues was asking. Okay, so if you want to use this time, is anyone still looking for group members? If you can just raise your hand. So if you're looking, those are the other individuals that are looking, and it's a great group. So I'm hoping that you will be able to communicate and find members and trade information, post on the discussion board.